And now we are going to have our first speaker, Joaquim Alves da Silva, who studied medicine here at the Universidade Nova in Lisbon, and is later after specialized in psychiatry. And at the moment, he's doing his PhD here at the Champalimaud Foundation, together with Rui Costa. And he actually fought to do his PhD in basic science because he thinks that in order to understand disease, one actually needs to know the fundamentals of it. And he is almost finishing his PhD, and we are really looking forward to his talk. Thank you.
I would like to thank, first of all, to the R committee, the people that organize R, which are amazing. I don't know how they find the time. And to the Marathon da Saúde also for the invitation. I spoke in the first R event, which happened 4 years ago. So I guess that's how long it took for them to forget how bad it was and invite me again. Not bad, 4 years. So I want to start by saying that scientists, basic scientists, they are haunted. But they are not haunted by ghosts. They are haunted by questions. Questions that wake them up at night, questions that in the morning they think they solved while they are showering and coming up with the perfect experiments that then they destroy on the way to work. And sometimes you never know how it's going to be because scientists are curious to the square. You never know which questions will attract their attention. Some questions are more easy to relate to, like why do we age, for example? Some others are not as easy to relate to. For example, why do unicellular algae move towards the light? Applied research, on the other hand, is much easier to relate to, like Daniel was saying. Applied research is similar to what we do every day. It's solving problems. We are very used to that. We are very used to think like that. Medical research is basically applied research. It's a noble way of research. It's driven by the urgency to help people. People that many times cannot wait. They cannot afford to wait for us to have the full knowledge, to know everything about the mechanism of a disease. So we need to work in a trial and error basis many times. And sometimes it works, but many times it's only works so far. Every physician, or almost every physician, will tell you that physiopathology is one of the main disciplines of medicine. And physiopathology has to do with understanding the mechanisms that give rise to symptoms and diseases. But physiopathology is only as good as our knowledge of the current system we are working on. That's why I want to share with you 2 stories today. These stories should highlight the link that there is between basic research and medical research. What we called here for this event, we called the invisible link. So first I'd like to tell you the story how the discovery of a neurotransmitter came up to be a major breakthrough in the treatment of one neurodegenerative disorder, which is Parkinson's disease. But also I would like to tell you how many times the link has the opposite direction, it's bidirectional. Sometimes clinics, medicine provides peculiar observations, so interesting that basic scientists jump on them right away, and they want to understand why is that so. And that leads to new findings, new questions, and so on and so forth. Okay, so as it's recommended, we should start from the beginning. And the beginning in this disease starts with James Parkinson in 1817. He described the shaking palsy. It was the first coherent description of Parkinson's disease, which at the time, he didn't put his own name to the disease, which is nice. He did, in fact, identify part of the symptoms, the tremor, which is a low-frequency tremor, the posture of the patients, which are curved, the rigidity also. But he called it a palsy. And afterwards, Charcot, by the end of the century, of the 19th century, he realized that Parkinson's disease, how he started calling it in homage to James Parkinson, was not in fact a palsy. Many patients did have palsy means like lower muscle strength. And many patients didn't have lower muscle strength. But he realized they had another symptom, which was bradykinesia, which is one of the main symptoms of Parkinson's disease, together with rigidity, postural instability, and tremor. And bradykinesia means slowness of movement, which can be due to the lack of initiation of movement or to the slow performance of the movement. The best way to illustrate it is to show you a video. In this video, you're going to see a patient. This is a severe patient. It's a published case. You can see it in YouTube in the New England Journal of Medicine. And it has a severe form of the disease. You see the tremor, you see the rigidity. The arms don't go around, don't walk, right? And the tremor is very gross and very obvious, and it can hardly move, it can hardly initiate. You see how the physician is tricking the patient into stepping by putting her foot in front of the patient. We're going to go back to that because that's an important observation. And there's the postural instability that we talked about. But let's be fair, right? Charcot and Parkinson, they did what they could. They described very well the disease. They were very good doing that. In fact, James Parkinson, when he described the disease, he did it from 6 cases only. And 3 of them, you only saw it on the street of London, never in his office. And 1 of them from far away. Still, he was able to identify the disease. That's remarkable. That's what happens when you don't have diagnostic tools. You need to really be a very good observer, right? And they didn't know anything or very little about, they knew something, but they didn't know a lot about the brain. Today, if you go around and ask neuroscientists, we all think we don't know a lot about the brain. At that time, they didn't know anything. So Charcot, Parkinson came up with some treatments to prevent Parkinson's disease, which were about bloodletting, like making wounds and bleeding the patient. And he even advised to put small corks on the wounds so they would infect and push away the inflammation from the central nervous system to the disease, to the wound. Charcot was more lucky. By trial and error, he found anticholinergics, which are used still today, but they are not very effective. One breakthrough needed to happen to start people thinking in that direction, and that one breakthrough, among others, came from Otto Loewi that got a Nobel Prize for it in '36. And it was the discovery of neurotransmitters. He did it in a very clever way. He stimulated the vagus nerve of a frog heart, and by doing that, he was able to slow down the heart of the frog. And then he just took the fluid around this heart and passed it to another heart preparation where there was no vagus nerve. And by doing so, he was also able to slow down the heart, which meant that there was something in the fluid that could target the heart that was released by the nerve. And that was the first neurotransmitter found. In the end, it turned out to be a neurotransmitter called acetylcholine. At the time, they started finding other neurotransmitters. One of the most popular ones was noradrenaline. As you can see here, you start with an amino acid, tyrosine. You have 2 intermediate products, dopa and dopamine, to reach noradrenaline, which at that time was thought to be a main neurotransmitter. Arvid Carlson, he started working on this neurotransmitter. And at that time, they had found this new drug, Reserpine. This new drug, what it did was once you injected in the animals that he was working on, rabbits, would decrease the levels of noradrenaline. And he found that the rabbits had a very strong behavioral inhibition and also symptoms that were related to Parkinson's symptoms. Then he thought, okay, this may mean that if I increase this noradrenaline by giving dopa, because I'll produce more, then I can revert the situation to this. So he gave dopa because from these substances is the only one that crosses to the brain from the veins. And voila, he got the result. The rabbits were no longer inhibited, they were moving normally. So he thought, now he went rushing, get the brains of the rabbits, look at the brains, and he was really sure that you're going to find an increase in noradrenaline afterwards, but he didn't. Noradrenaline kept low, and then that was a problem. That's an anomaly in the sense of the science of the epoch. And this anomaly left him with an heresy, which was to say that one of these 2 products was in fact a neurotransmitter. Nobody believed that at that time. And he had a hard time to prove it, but he did show it, because then he measured this neurotransmitter dopamine, and they found that in fact it was the one that rised afterwards. So this should be the one that is helping the rabbits to get better. At that time, Arvid Carlson also realized that people had already been describing places in the brain where the motor function that is disturbed in Parkinson's disease, or let me say it the other way, the people have found that there were certain brain areas, like this one, which is a striatum, which were important for function, the motor function that was disturbed in Parkinson's disease. So of course, one of the first things that Carlson did was to study dopamine in this part, and he found exactly that dopamine was decreased in the striatum. Of course, clinicians noticed that, and they went to the brain of Parkinson's disease patients, and they found exactly the same thing. They found that dopamine was lowered in this part of the striatum. Furthermore, they also found that cells that were missing here in Parkinson's disease were in fact dopaminergic cells, cells that produce dopamine. Nobody knew at the time that they were doing that. And that's a part of the brain called substantia nigra compacta. And then they realized that it was in fact the neurons that were here that were projecting to the striatum and feeding the striatum with dopamine. And of course, you are thinking probably what they thought at the time. Okay, if the rabbits get better with dopa, maybe I can get patients better with dopa. And it was very fast. From the discoveries of Carlson, it took like 1 or 2 years for people to try it in Parkinson's patients. And I cannot have more impact than reading the words of the original authors that tried dopa for the first time in patients. Bedridden patients who were unable to sit up, patients who could not stand up when seated, and patients who, when standing, could not start walking, performed all these activities with ease after L-dopa. They walked around with normal associated movements, and they could even run and jump. So this meant that now there was a paradigm shift. There was a new neurotransmitter, which was a fundamental neuroscience finding that then enabled clinicians to click and find the treatment, the first appropriate treatment for Parkinson's disease. And the cool thing is that this treatment, although it was found, you can see, almost 60 years ago, it's still the main treatment for Parkinson's disease. I told you that I wanted to tell 2 stories. This first story illustrates this link. And it's really an invisible link because it's much easier to relate to the treatment discovery than to all the basic research that was done that led up to it. Maybe not in this case because Carlson did get the Nobel Prize in 2000, so he was recognized for that. But I also wanted to tell you about the bidirectional of this link. It's a bidirectional link. So I want to show one thing. Remember the patient I showed you in the beginning? So what happens when we sit this patient in a bicycle and we push him? It's the same patient. It's the same patient you saw before with the same problems, same symptoms, but when we push him on a bicycle, he can ride the bicycle. You could not say it's a Parkinson's patient. At a certain point, he can even turn the head without problems. He turns, and at some point you're going to see that he gets up from the seat and he's pedaling to get more power. So this is incredible. Every time I see this movie, I think it's amazing. And it sparked the hypothesis at which I worked in my PhD. So now I'm going to tell you about how clinical observations inspired the basic science that I did during my PhD. So we thought maybe there's something. What does this tell us? This tells us that the lack of dopamine affects movement. We know that in a very serious way, but it's not all movement, not all the time. But as soon as he stepped out of the bicycle, he's froze again. It's amazing, right? But it's not all the time. There are situations where movements are okay. And this meant for us that dopamine could not be related to all moments of movement. We thought that maybe dopamine was related specifically to the initiation of movement. But even working with animal models, it's a challenging hypothesis because we need to be in control of the neurons. We need to be able to activate these dopamine neurons or inhibit these dopamine neurons when we want, because it's the only way we can explore if the dopamine neurons are important for the initiation or for the ongoing movements. Okay? Luckily for me, when I was joining the lab from a few years, I've been running a revolution in neuroscience, which is called the optogenetic revolution, one that Susanna Lima, one of the researchers that work here, was also part of. And amazingly, that revolution had a lot to do with one of those stranger questions. It had to do with researchers that wanted to know why a green algae is able to move towards the light. How does that happen? How can she swim towards the light? And then they found that, in fact, it was because of special proteins that they are called rhodopsins. These rhodopsins, when they see the light, well, see is a strong word, right? When they are exposed to the light, they change conformation, they open, and they behave like ion channels. These ion channels change the current of whatever cell they are on. In the algae, they move a flagella, but in neurons, maybe they could make them fire. And that's what people thought at the time, and specifically, Ed Boyden and Carl Dijsselrath were the first to express this kind of proteins in mammalian neurons. And they were able to drive those mammalian neurons for the first time with one thing which is very important in the brain, which is a fast time resolution. But there is no better way to then show you how this happens. So what I'm going to show you are recordings. In fact, these are recordings from dopamine neurons, ones that are lost in Parkinson's disease. And these are recordings which were done when the mice were moving around, okay? They were not anesthetized, they were moving around, behaving as they do. And how do we do this? We put an electrode in the brain. You can imagine an electrode to be like a microphone, okay? It's a microphone, it's next to dopamine neurons, so you can hear the neurons talk. The neurons talk in electricity, okay? Also, we know that with neurotransmitters, but we can listen to the electricity when we use electrodes. What you're going to see, you're going to hear also some popping sounds. This popping sounds means that the neuron is firing, and you're going to see this upward deflection in the traces. It's when the neurons fire. I'm going to show you a regular tracing, and then I'm going to show you one which is being controlled by light. In fact, two. One which is activated and the other one which is inhibited. Okay, so what happens if we express one of these proteins and we shine briefly light, like 1 second with a 1 second interval? What we get is that we make this neuron talk. You saw now the neuron fired with a pattern. We could predict when it was going to fire because we knew that after 1 second there was a light coming on. So now we were in control of this neuron. But also we can do the opposite. Certain proteins allow us to inhibit the neurons and silence them. And the microphone does not pick any sound because neurons now cannot speak because the current, the changes don't happen that allow for them to fire. Okay, now that we could do that, we were obviously not the only ones doing this kind of things. Everyone was at a certain point started doing this, and this allowed us to probe the brain in a very specific way. We could know which circuits do, how they communicate with each other, what happens if we take a part of a circuit out, and what happens to the behavior of animals. This was a major breakthrough. In the case of our work, we used it for the hypothesis I told you about. So basically, we had a mouse moving in a square, and when it was not moving, we activated these neurons. Sometimes we activate it, sometimes we don't, and then we compared. And we found that if we inhibited these neurons first, we avoided the neurons, we avoided the movement of the mouse. You see, if we don't inhibit, the mouse can start moving after he's immobile, but if he's immobile and we inhibit his neurons, he cannot start moving. But if he was already moving, and we inhibited these neurons, then nothing seemed to happen. And on the other hand, if we activated these neurons and the mouse was not moving, we could promote movement of the mouse. But if he was already moving and we activated these neurons, we couldn't really change his behavior. So what the results of our work show is that somehow there is a specific role for dopamine in the initiation of the movement, which is not that important once the movement is already going. Okay, so that's it. In respect to what we did, I just wanted to finalize with 3 main ideas about what I talked to you today. And the first idea is that fundamental science, basic science, fuels, in fact, humanity with knowledge. In case of medicine, that knowledge becomes healing power, and we can use it to improve our understanding of diseases and come up with better treatments, better diagnosis, prevention of disease. Medicine also fits basic science with interesting and peculiar observations that allows us either to test new ideas, to test new questions, or even to question older ideas. And for centuries, this interplay has been happening, even though it's invisible, like the title of our meeting suggests. It has been happening. It's what has been pushing medical progress also. And at times I wondered, while preparing this talk, if it was really necessary to talk about this. It's kind of obvious, maybe because I work in an institute where basic science is happening. And then I recalled what happened when I told my mother that I was going to leave the hospital. And then my mother asked me, what about the patients? I said, well, the patients, yeah, the patients, they need you. I said, oh, of course. But I didn't know what to say. Luckily, my wife said, who knows, maybe you can figure something out that helps more patients. And then I knew everything would going to be all right. That's it. Thank you, man.
So thank you, Joaquim, for your really nice talk. And now there is some time for questions. If someone has in the audience, please just raise your hands and you can question Joaquim. There is some microphones. So maybe I start with one. I would like just to ask you how difficult or easy was it just for the scientists who wanted to describe dopamine as a neurotransmitter, but all of them thought that noradrenaline was the end product. How easy or difficult was his task to convince the community that actually was dopamine doing the job?
So that's an interesting story. Arvid Carlson told that story in his Nobel laureate talk. And it turns out that after one amazing thing is that the paper that describes dopamine as a neurotransmitter is 1 column long. And I don't know, maybe you are not related to this, but nowadays papers are very long many pages, and sometimes too many pages. In 1 column, he described something that changed neuroscience forever. But once he talked to his colleagues about it in the first meeting he went, they didn't believe them, because there was this eminent figure, Henry Dale, which is very important for neuroscience, also for the discovery of neurotransmitters, and he didn't believe that dopamine was a neurotransmitter. And Arvid Carlson basically gave up on that meeting, and people didn't even mention his work during that meeting. And people carried on behaving as normal, thinking that noradrenaline was the only neurotransmitter, and dopamine was a byproduct of that pathway. Years later, 3 or 4 years later, after all these things have been described in Parkinson's patients, etc., then eventually it turned around, and people started believing that dopamine was a neurotransmitter. But that took time. It's incredible, because it took more time than the time it took for physicians to test dopa in patients. And sometimes that happens in science. It's tough to change a paradigm. A revolution needs to happen, and revolutions are never easy.
Thank you. Any other questions from the audience?
Thanks, Joaquim. Very nice. If I may say, like your mother. Now, So I understand very well that if you are a medical doctor specialized in diseases of the nervous system, that you come and do research in nervous system, then of course it's very helpful for your medicine. But don't you think it would also be helpful, even if you not come to do research on neurosciences, but if you do research on cell biology or something? Would it also be useful, and why?
I think it would be not useful. I think it would be something I would do. It's not neuroscience that drives me. It's I don't know. It's the human endeavor of being part of science. It's trying to discover things. I could do that in neuroscience. I don't know if I'll be good at it, but I would like for sure to do that as a way of living. And I don't know, I have this feeling that it's bigger than we are, right? It's something bigger than us. It's the human enterprise. So we can, and being part of that could help anybody, a physician especially, but it could help anybody. That's why we have all sorts of people working in the lab.
Thank you. You were saying it took more time to prove that DOPA was the working substance than to use it in patients. Today, it seems that it's the contrary. How long would it have taken today? How come L-DOPA was given to patients so fast? Was it without knowing if it was safe? I mean, I think it was very good not to have to wait, but was it already known that it was not going to do them anything?
I think that, like I said, medicine is driven by a sense of urgency that's different from many other aspects of what we do. And even because of that, we have rules in place that avoid us to make mistakes, right? Like clinical trials, proper clinical trials, all the phases of until you get to a drug, right? And that's very important because there were a lot of mistakes, of course, down the line. Even what I told you about this, DOPA was first injected, and then it had also side effects, and then another physician, important physician, figured out that oral regimen that with increasing dosage would be more effective and would free people from getting an injection, and that took time also. But I think, to answer your question, my explanation is because of this sense of urgency. It's because it's very hard being a physician and facing people who need you and you don't have anything to offer. And then you try sometimes you try whatever you can. And in that case, it was a very compelling evidence that it would work. But I totally agree that we should be careful when we pursue this, because sometimes we get blinded by this drive to help, and we make mistakes. So that's why everything is in place to avoid that.
Hi. So we're trying to figure out how to connect basic research with clinical research. What difficulties did you find coming from a medical background and going to research? And what difficulties do you anticipate in trying to translate the work that you've done as a researcher back to clinical practice? And how do you think that we can maybe try to make it a more regular thing, instead of being an exception?
Yeah, that's a great question. So the first part of the question, the transition, for me particularly, I think it was easy because I found great people here. It's an amazing institute. The students are very helpful, and it's a great place to work. So that part, I think it was easy. Also, there are things that a physician has which are helpful. Like we have, like the schedule is very tight. It's long hours, right? And urgency, the ER, and that also allows us to maybe in the first part to be more resilient to the effort of the start of the research. But most of all was because of the help of others, like Fatuel Teco Petla, a lot of people, Rui Costa, which is my mentor, everyone in the lab, the other labs. It's an incredible environment. So that part was easy. The other part is much tougher. I don't like when people try to push their findings into clinical just because. And I don't think there's a real direct application. We can say something like, we can say all treatments right now for Parkinson's disease are focused on sustained treatments. And one thing I didn't show you is that we also found that there is transient changes in dopamine neurons which are related to this initiation of movement. So people may start thinking, people already think like that, but it's hard to change from sustained treatment to phasic treatment, or like how can I explain this? Instead of having a drug working all the time, or a stimulation, like deep brain stimulation working all the time, have it on a kind of a demand. So I could speculate that, but to be honest and to be fair to people that are really doing clinical research, there is no clear correlation. There is no clear association to clinics. But that's okay. That's what Professor Coutinho was saying. I don't need to be directly related to clinic to understand and to be a better doctor after this. It's the experience that counts.
Thank you.
Okay, thank you very much for your questions. Thank you, Joaquim.